Hello again. So far we've been concentrating our look at genetics on recessive conditions. And so we've been looking at sex-linked and autosomal recessive conditions. What we're going to look at in this session is the inheritance of conditions which are dominant. And to do this we need to just free ourselves of any preconceptions and to some extent turn our thinking inside out.
Neurofibromatosis is a very serious condition which is inherited in a dominant fashion. It isn't sex-linked. So in other words, the disease allele can be shown on this diagram as big N, and the recessive allele is the normal unaffected version of the gene, and we'll show that as small n. You can see from the key that it's only double recessives for small n who avoid the condition. Just possessing one large N is enough to guarantee that you will suffer from the condition. When solving a pedigree diagram for a dominant condition, exactly the same principles apply that you've encountered before. So to start with, we'll identify all of the known recessives as they are clear of the disease and therefore have an unaffected phenotype. Because all of these unaffected children must inherit one gene from each parent, we know therefore that the affected mother must be a heterozygote. But can we be sure of the genotypes of the affected children? Yes, we can. They must all have inherited the single disease allele big N from the mother, and therefore can only inherit the normal recessive version of the gene from their dad. So to sum up, with dominant conditions exactly the same principles apply, but you just have to free yourself of the idea that dominant is good and recessive is bad.
So far we've been working with single genes with two or more alleles, but quite often we may be interested in the effects of more than one gene. So to finish off this series of screencasts, let's look at how we can apply the Punnett square to two genes, each with two alleles. We're going to take, as an example, the eye colour allele B, which has got the dominant form big B for brown eyes and small b for blue eyes. For our second gene, we'll use an old friend. We'll use the gene for earlobes. And so we're going to use capital E for free earlobes and small e for attached earlobes. Now, let's imagine we've got a couple who are each heterozygous for both of the genes. In other words, they're big B, small b for eyes and big E, small e for earlobes. Now, in order to accommodate the extra set of genes, we need to use a bigger sized Punnett square to accommodate the extra gene and allocate it to gametes. As we did before, when we separate the alleles into the different gamete combinations, we'll therefore need an E and a B each time. So what we're going to do now is to go through it. So just to remind you, our original cross is big B, small b, big E, small e, crossed with the same. We're going to place one of the partners along the top axis, just as we did before. So big B, small b, big E, small e. And this time what we need to do is we need to take one of the big B alleles, place it in that box. So we'll take big B, one of the E alleles, we'll take big E and pop that into that box. Next box for gametes, big B and small e. Next one's going to have a small b and a big E. And then we've got the two recessive alleles going into the final gamete possibility. And the rows down the side will actually be the same because we're crossing two heterozygotes. So just fill those in. And then we need to start to work through the columns. So we'll go through column 1. It's the big B column, so we'll pop a big B into that first cell. But it's also the big B row, so we'll add the big B there. It's the big E column, and it's also the big E row. That's the first possibility at fertilization. Next one down, again, big B column, big B row, big E column, small e row. Next one, big B, small b, big E column, big E row, etc. Now what we'll do is we'll work our way along the top row just to complete that little part of the process. So this is now the big B column, big B row, big E row, small e column. Notice that we always put the dominant, the big letter first before the small. So big B, small b, big E, big E. And then finally, one just like mum and just like dad. So we'll just carry on and complete the table now. Just got 2 more to do. And the final one, bottom right hand corner, we've got the double recessive. And all looks very confusing, but what we can actually do is go through that table, that Punnett square, and if we start to pick out the ones with the various combinations, for instance, big B's and big E's, we know that any of them with big B's and big E's are going to have brown eyes and free earlobes. And if we do that, then what we find is that we get a very characteristic ratio for the cross between 2 heterozygotes. What we actually get are a ratio for the individuals with brown eyes and free earlobes. We find there are 9 out of 16 of those. For the individuals with brown eyes and attached earlobes, which is a recessive characteristic, we find there are just 3 of those. Individuals with blue eyes and free earlobes, again, we've got 3 of those, 3 out of 16, and just 1 out of 16 is the double recessive with blue eyes and attached earlobes. So you can see that in a ratio sense, 9 out of 16, there's a ratio of 9 out of 16 with both dominant characteristics. 3 out of 16 for any of the recessive characteristics, and 1 out of 16 actually for the double recessive with blue eyes and attached earlobes. So that's how you do a dihybrid cross using a Punnett square. Always remember that it's a ratio expressed out of 16, not out of 4, as we did with monohybrid crosses.